Jeremy Ferrier. I'm the head of products at Iconeus, and it's my absolute pleasure to be here today and talk about functional ultrasound neuroimaging and a little bit of what we do and develop at Iconeus. So without any further ado, this is the outline of my presentation today. I'm going to start by explaining the basic principles behind functional ultrasound and how does it work. And then I'm going to give you an overview of the FUS research through case studies and published studies. And I'm going to end this presentation by showing you the Iconeus One imaging platform that we developed at Iconeus, which is a solution for functional ultrasound for neuroscientists. So when it comes to brain imaging modalities, there's a lot of techniques that are available in the neuroscientists' toolbox. They all have their strengths and weaknesses. So first, fMRI, which is the gold standard, has an excellent penetration and a good spatial resolution. We uses bold signals to probe the brain activity in 3 dimensions, but it's rather slow and lacks a little bit of sensitivity. PET imaging has an excellent penetration, but a rather poor spatial and temporal resolution, but it has an excellent sensitivity due to the radio tracers. On the other hand, optical imaging has an excellent spatial and temporal resolution and an excellent sensitivity, but it is restricted to the surface of the brain and cannot image the deep structures, subcortical structures. And one of the imaging modalities that have been quite overlooked in neuroscience is Doppler ultrasound, where you can visualize blood flow through the Doppler signal. Ultrasound is one of the best techniques to image deep in the tissue with an excellent sensitivity, spatial resolution and temporal resolution. But the main issue that is it lacks sensitivity. As you can see on the image, you can actually only see the major arteries of the brain. And to overcome this sensitivity problem, functional ultrasound was developed with the first publication in 2011. And it's actually one of the best techniques to image the microvasculature with high sensitivity and a high resolution. So how does it work, and what is the difference between functional ultrasound and conventional Doppler ultrasound? So both techniques relies on the use of a linear transducers that is positioned above the head of the pulmonal with ultrasound gel for the acoustic coupling. And conventional Doppler on the left is based on focused beam that scans the brain line by line, one after the other, while functional ultrasound that you see on the right uses plane wave unfocused ultrasonic waves that are sent at ultra-fast frame rates. And this results in a 100-fold gain in sensitivity. As you can see on the left, you can only measure like the bigger vessels, but on the right you have access to the smallest and the slowest vessels of the brain. If we look at it like along the time dimension and look at this image formation, it's giving you like the striking difference that you see in terms of pixel density, which is like the amount, the time signals, the data points that you get. With conventional Doppler on the left, the amount of the pixel density is restricted by the time takes to form one line of the image. FUS on the other hand, because it's using plane wave unfocused, actually has like a higher pixel density over time, given like the same pulse repetition frequency. And this is the key to get this boost in sensitivity. So you get with functional ultrasound a longer signal and also a lower noise and a lower background. And this gain of sensitivity, this access to the smallest vessels, it's actually fundamental for brain activity mapping. Let's zoom in on the microscopic level and see what's happening inside the brain. So the signal that we measure with FUS is actually directly proportional to the cerebral blood volumes, which is the amount of red blood cells coming for the pixel at the time. So when the neurons get active, there is an increase of vasodilation of the neighboring vessels, which results in an increase of the amount of red blood cells. And you can see on the plot, I cannot point at it, but you can see an increase of the signal here, which is what we call a hemodynamic response. And this phenomenon is called the neurovascular coupling, and this is the basic to probe brain activity through the blood flow. Now, one of the nicest and easiest examples that we can give is a whisker stimulation. This is the things that we love to demonstrate with our device. So if you position the probe above the brain, a gentle deflection of the whiskers results in an increase of the CBV locally in the cortex, in the barofield cortex, which you can measure in real time, and then you can make a correlation and basically map which brain regions were active during any type of task or any stimulus. So just to give you a few numbers about functional ultrasound, you get a 100-fold gain in sensitivity compared to conventional ultrasound. We can capture blood vessels with velocities down to 1 millimeter per second, which corresponds to the smallest arterioles and veins, so pre-capillaries. We get an excellent spatial resolution, 100 microns in plane, we're at 15 megahertz. And we get a sensitivity that is high enough to detect hemodynamic changes in a single trial. So a typical sensory stimulation like the whisker stimulation that I just showed results in a 20-30% change of signals compared to rest. And we can even detect CBV changes as low as 2% compared to baseline. So that's how sensitive the technique is. So I'm going to present a little bit the major application for functional ultrasound in neuroscience, which can be categorized in like three major applications. The first one being task-based functional imaging, or what could be understood as stimulus-based activity mapping. Again, another example of whisker stimulation that I just showed, but this time in mice, and that's the good opportunity for me to say that functional ultrasound can be done in mice completely non-invasively through the skull and the skin. And you can see again like the activation in the cortex and the thalamus. This second example that I like to show is an example of epileptic seizures, and that's actually coming from the very first paper on functional ultrasound from 2011, where they injected a 4-AP in the cortex and trigger an epileptic seizure basically. And as you will see on the video, we can actually map a spreading depolarization that propagates along the cortex and also on the other hemisphere and subcortical regions. And these seizures are chaotic by nature, so this is the only technique that allows us to visualize these seizures in the whole brain at this level of sensitivity, because we cannot average this kind of events that are chaotic. This third example is about visual stimulation, so just placing a rat, an anesthetized rat, in front of a monitor that is actually blinking at defined times. With functional ultrasound, you can map the whole visual system from the cortex to the LGN and superior colliculus. And what you can see on the plots in the right is that every time that the stimulation is presented, you see this increase of the signal that is up to 10%. And I like to show that to show a little bit the sensitivity. Even with the first trial here, we can actually detect that the region is activated. So we don't need to average a lot of trials to have a complete map of the activation. Now, these three examples were performed in anesthetized animal, but one of the main advantage of functional ultrasound is that it's actually compatible with awake and behaving animals, because we work with very small transducers that can be tethered to the animal's head. And for that, in mice, we just need to implant like a small head plate and leave the skull intact. And then we attach the probe through a magnet probe holder. So this next video is going to show you how easy it is actually to plug the probe to the animal. You just need to apply a little bit of ultrasound gel to the head and then plug the probe, adjust a little bit, and then you can have access to the brain activity in real time whilst the animal is actually behaving in its home cage. So it's very, very powerful. The same strategy has been applied to rats. Here it's a nice example where the animals were trained in a corridor going left and right and get a reward at the very end of the corridor, whilst we imaged using functional ultrasound and also with EEG recording with LFD. And You will see on this video here on the left, this is like a depiction of the rat's trajectory during the experiments. Here you have the functional ultrasound data, and on the right you have the EEG, LFT recording, and also like the speed and accelerations. And what you can see is that at the beginning of the task, when the animal is learning the experiments, the activity is mainly restricted to the cortex, but very mild actually in nature. But as the animal is repeating the task, you will see here in the intermediate run, the dorsal hippocampus is actually more and more recruited, and you will see here that the activity, the change, the hemodynamic response gets restricted to the hippocampus. And it's even more striking towards the end of the experiment when the animal have done that 100 times. Like it's switching almost to like a robotic mode, not like recruiting the cortex anymore, but just doing the task without too much thinking. So this is a nice example on how you can decipher like the brain activation pattern across a whole coronal section with a natural behavior. In another studies, like these authors went a little bit further, and imaged the rats during sleep with the same system, hoping to see that this pattern of activation will repeat. But what they show instead is that during a specific phase of REM sleep, there were this vascular surge, this huge increase of blood flow in the brain that is widespread and coming with waves. This is a phenomenon that was described in humans, but that was the first time that I know that it was described in animals, which explains that there's probably an evolutionary role for this phenomenon. But actually it's very hard to make an animal sleep in an MRI magnet because it's so loud. So here another example of natural behavior and brain activity recording related to this. Functional ultrasound is also very useful if you're interested in pharmacological challenges, and if you're working and studying a drug and you want to know which brain regions are activated by the drug. You can see here, it's an example of donepezil injected in an anesthetized rat. So provided that the drug itself doesn't have like a vascular effect, if you see any change in the CBV before and after the injections, that's most likely explain that there's like an activation or an inhibition of these regions that you can track over time. That leads us to the second major application of functional ultrasound, which is functional connectivity, and it's like a huge domain in neuroscience. So functional connectivity is defined as the temporal correlation between two distributed neuronal events happening in the brain. So these first studies in 2014 just looked at an anesthetized rat and just recordings for 10 minutes. You can see that the signals here is not completely flat. Actually, you have like oscillations of the signal, and this is not random. If you look at the somatosensory cortex here in red and the other side in blue, actually the signals coming from these two regions are almost perfectly in sync. Like they co-fluctuate over time. On the other hand, if you take the green regions, the cingulate cortex, and compare it to the red regions, you can see that these two plots are not in sync. So technically, when you're looking at the brain at rest, the regions that are functionally related and connected to each other, the signal tends to be synchronized over time. And if you extract the signal from all the brain regions you can identify on the image, you can build this kind of connectivity matrix that you see on the right, which is actually like a barcode of the brain health. It gives you information into how the brain is organized region by region. So you can identify networks of regions and how the dynamic change in health and disease. And technically, in a lot of neurological diseases like Alzheimer and depression and schizophrenia, it has been shown in humans mostly that the functional connectivity is impaired, and sometimes before even like clinical symptoms can be detected. So it's like a biomarker of the brain organization. What we did is, that was one of my postdoc projects, is to look at the effect of anesthesia on functional connectivity. And for that, we compared these correlation matrices in animals that were awake, mice, head fixed in the mobilum cage, and look at the different level of sedation, like using medetomidine, which is like a classical anesthetics. And what we saw is that functional connectivity is actually impaired with by anesthetics in a dose-dependent manner. So if you're interested in mapping functional connectivity, it's important to go awake, because the awake brain is, you don't have the anesthesia bias, and functional ultrasound is the way to go. Now, everything that I showed so far have been done in 2D with linear transducers, so you can capture several slices at the same time, but to cover the whole brain of a mouse, it would take like 10 seconds, which is a little bit too slow for functional connectivity or even for activation mapping. But at Iconeus, we developed a couple of years ago new transducers that we call multi-array transducers that allow the simultaneous multi-slicing of the brain. And just with like a few steps of the probe, you can cover the whole mouse brain in about 2.4 seconds, which is fast enough for functional connectivity. Actually, it's perfect for that, and also for any type of activity mapping. So these transducers are compatible with anesthetized, head-fixed, or awake head-fixed animals. And we explore the functional connectivity in awake mice. And now you can reconstruct this kind of correlation matrix over 200, 300 brain regions that are extracted from the Alien Brain Atlas. And you can go a little bit beyond and use unsupervised network identification, like this type of decomposition, like ICA or independent component analysis, that extract for you almost automatically like the brain modules and the brain networks, and separate them. And we can identify like the DMN network, the deformal network, which is kind of famous because this is the network that is almost consistently firstly impaired in neurological diseases. So it's paving the way for deciphering like how the brain networks like change with genetic diseases or using pharmacological agents. Now I'm going to talk about like the last application, which is vascular imaging. As you may have noticed with functional ultrasound, we have a very clear, detailed map of the vascularization. Actually, when we see these images, people tend to think that this is micro CT or other type of technique, but it's really ultrasound without any contrast agents. So doing this kind of scan of a rat brain takes a few seconds. So you can identify like the major arteries, the major blood vessels in the brain. This is what's been done in the rat here. Using a tomographic approach, you can even reconstruct a 3D volume with isometric resolution of 50 micrometers. And you can extract the vascularization from region of interest from the atlas and then see how that changes over time, doing like a follow-up and longitudinal studies. So this provides like a good overview of how the vascular system, the vascular tree is organized in the brain. And it's actually really nice, but we can do much better than this. And we can do that with ultrasound localization microscopy, or what we call super resolution, FUS. It's kind of a sister technique from functional ultrasound, but this time this is based on the use of contrast agent. And to explain a little bit how it works, I'm going to show you a video of the Eiffel Tower, because it's very close from home. And as you may know, the Eiffel Tower is actually blinking at every hours during the night. And let's imagine that you have a camera that is pointing towards the Eiffel Tower. You can see it blinking. If this camera has a very high frame rate, you will be able to separate the spots that are blinking and are very close to each other. You will be able to distinguish and pinpoint them very precisely. And as you accumulate the data, you will be able to reconstruct a very detailed, super resolution image of the Eiffel Tower on the right. And we can do, so this is optical, but we can do the same thing with ultrasound. And what we use instead of blinking spots, these are microbubbles and gas microbubbles that we inject intravenously And this provides contrasts for the ultrasound, but we can also detect them and track them with very high details. So this is a video that's showing a rat brain. You can see all the microbubbles detected and all the tracks accumulating. And you know, as we accumulate data, it starts to draw the vessels in the brain with very, very high details, almost down to the capillaries. So with this type of techniques, we go down to 5 micrometers resolution. And this is the type of images that you can get across a coronal section of the rat brain. So when people first see these images without context, they tend to think that this is microscopy, that we just slice the brain and then stain the vessels, but this is actually a living rat brain. And so this animal can be then wake up and then re-image the next day, so you can do a follow-up. But that's not it. You can also get access to the velocities of the blood at the single vessel resolution in millimeter per second. And you can have access to the direction. And this is what this image is showing right now. So everything that is blue is going up, and everything that is yellow is going down. And if you zoom in the cortex of the rat, that's allowing you to distinguish between actually veins and arteries. Arteries are going down, and veins are going up. And then you can quantify a lot of things from this rich data. So of course, it's very interesting, but people were interested in having like the whole brain in 3D and not just a coronal section. So we developed a new type of probe that is called RCA, for raw column address transducers, that allows you to capture volume at the same time instead of just a cross section. And we can apply the same thing and get access to the super resolution map of the vessels, this time by tracking the microbubbles in every direction. And of course, this is non-invasive. So this animal here, it's a mouse, can be waken up and then re-imaged several times. So if you suspect that there's a vascular plasticity in your model, this is the best imaging technique for vascular imaging. Of course, from this data, you can extract a lot of parameters, like the morphological parameters from the vessels. We can also extract the blood flow from it, and you can like make this kind of graph when you can see, you know, region by region, how this areas account for the total CBF in the blood and how that changes across genotype, across diseases. And also you can do this type of single vessel analysis as well. So I'm going to finish by talking a little bit about Iconeus 1, which is the functional ultrasound system that we developed at Iconeus, that you can see here. And it's not only elegant and very beautiful and a nice aesthetic addition to your lab. This is actually the only full-featured commercially available functional ultrasound device that you can get. It comes with a motorized robotic platform that allows you to scan the brain of any animals that is head-fixed and positioned under it. And you can do that from the software inside. And of course, it comes with a range of transducers with high sensitivity and tailored to different application. But that's not it. Like if you look inside, you're looking at a complete software suite that is actually get you covered from the acquisition with a real-time visualization to the analysis with an automatic atlas registration, extraction of the data, and also a very powerful 3D visualization. Of course, it's everything that I've showed can be done with the system, and you don't need to have like any background in ultrasound or programming. You don't need to understand how the ultrasound works. You can extract data really easily with the system. It's really easy to use. It's compatible with awake imaging, and also with a wide range of techniques that you find usually in the labs, like EEG, optogenetics. Everything can be combined to extract data and cross-correlate different approach. So we are at booth 613, that is actually right across 10 meters from here. So please stop by and come. There's a lot of neuroscientists that are here to answer any question you may have, myself included. We also have a lot of goodies. We have a nice logo. We tend to print it many things. So if you want a bag, if you want a mug, please come by. Thank you for your attention.